Hello all and welcome to chapter 4. We're going to call this part A. So today what we're going to do is we're going to be talking about DNA and genetic material. So first of all, we're going to do a little bit of review. When we think about this review, I want to think back to chemistry and how we talked about those macromolecules that were organic. So we talked about proteins, for instance. They are organic substances, meaning that they contain carbon and hydrogen. But when we talk about proteins, we want to know why they're utilized in the cell. So they're used as structural materials for our channels, for our membranes. They act as an energy source as well to be actually a carrier molecule. We have them derived as hormones and receptors. Another big thing is that they're actually useful as enzymes. And enzymes are a biological catalyst that help to speed up chemical reactions. We're going to be talking a lot about enzymes coming up, but I just wanted to make mention of that. And they also serve as our antibodies as well. So proteins are kind of the workhorse of the cell. And when we think about their building blocks, remember that they are made up of amino acids. So when we think about an amino acid, we can see them down here at the bottom. They all are going to have a central carbon atom. And so when we look here, I'm going to draw the central carbon atom, you'll see that there's a carbon attached to a hydrogen. It is also attached to a carboxyl group, and it will be attached to this amino group. And then the fourth bond to this carbon is the R group. And the R group can change, and this is what gives the different unique characteristics to the amino acid. So they all have the central carbon, the hydrogen, the carboxyl group, and the amino group. But the R group is very unique. So we can see with cysteine how it contains that sulfur. And then we can see with the phenylalanine how it has this actually very complex group where we're able to see these carbons actually associated in a ring. You don't have to know the different types of amino acids, but I want you to know that they are the building blocks and they build up proteins. And I kind of want you to know somewhat of what they look like. So that was just a little bit of review to keep in mind. Now, when we talk about actually putting these amino acids together, we string them along in something called a polypeptide chain. And what you're going to see is that they come together via this bond between the carboxyl group and the amine group. Again, not going to ask you any detail about that. I just want to make mention. So when we talk about stringing amino acids together in order to make a protein, you kind of have an idea of chemically what's going on. And then when we think about the levels of the protein structure, what we start off with, again, at this primary group is the amino acid sequence. So the amino acids are placed in a very proportional and very specific sequence in order to actually build up this protein structure. The structure greatly relates to the function. So at the primary level, you're just seeing this amino acid sequence. Then finally at the secondary, we're going to be seeing this kind of twisted structure, if you will, and that's because of hydrogen bonding. So there's those little weak forces that are coming together and they're forming these bonds. And then finally, when we hit the tertiary, again, we'll see a 3D structure and we'll see this true folding of the protein. And then when we have some of these proteins actually come together, they're going to build this giant protein in a quaternary structure. And again, that's when we really see the very specific shape that's going to relate to its function. So when we talk about building up proteins, again, we have to start at this primary level and have our correct sequence of amino acids, and then eventually run through this series to build up this quaternary structure and make our specific one protein. Now, also, again, just a little bit of review is talking about nucleic acids. So nucleic acids, we're going to think about as our DNA or our RNA. DNA is deoxyribonucleic acid. RNA is ribonucleic acid. And both of them carry this phosphate sugar backbone. For DNA, it's deoxyribose. For RNA, it's a ribose sugar. And what you're going to see is that this building block of nucleic acid are called nucleotides. So again, they have this phosphate sugar backbone, and you'll see that they have this nitrogenous base or organic base to them. And when we think about the function of DNA, it's to help to carry our genetic code. We have a very specific blueprint that gives the instruction manual of how to make proteins and how we make our enzymes in these channels. And then RNA is going to aid in actually building and making those proteins. So having that amino acid sequence all the way up to, again, actually that building of that quaternary structure. So when we talk about, again, these two major types, I want you to know DNA and RNA. DNA, again, is going to be double-stranded, and it's double-stranded in what we call a double helix structure. So they kind of form almost this ribbon chain, if you will. Again, they have a phosphate sugar backbone, and then in between are base pairs. And we're going to talk about these base pairs. We have A and T that go together and C and G that go together. So we have adenine, guanine, cysteine, and thymine. And so the way I used to remember this is that my name was Alyssa Thomas. So A and T went together. Now I got married, and of course my last name's Cooper, but still remember my maiden name is Alyssa Thomas, and that'll help you to remember which ones go together. But again, they're made up of these nucleotides, and DNA's whole goal is to store that genetic code. And where is DNA stored again? In the nucleus. So I hope you remember that. All right, now when we think about RNA, RNA is going to contain a ribose sugar. And the DNA helps to store the genetic code, but we have to actually turn that genetic code into a message that can be conveyed to make proteins. That's where RNA comes in. It's going to be single-stranded, not double-stranded, and its nucleotides are going to be a little bit different. We still have adenine, guanine, and the cytosine, but instead of having thymine, we use uracil instead. So I do want you to know the differences between DNA and RNA. All right, now we're going to get into the new stuff. So I hope that was a good little review. Keep that in your mind as we're going through this next chapter. So the first thing that I'm going to talk about are enzymes. And so enzymes are actually a type of protein. We call them a biological catalyst or a protein catalyst. And what they do is they help to speed up chemical reactions. They increase the rate of chemical reactions. And so what they do in order to make this occur is they lower something called the activation energy. So really quickly, let me fix my screen. And so imagine, if you will, that in order to have lactose be broken down into smaller components, the activation energy to make that happen would be very high. The energy required to make that reaction take place. So because of this, this is a very high little hurdle to make that happen for our activation energy. And usually we show it as this. So what the enzyme does is it helps to lower the activation energy. So when you use the enzyme, it helps to lower that activation energy and make that reaction happen a lot faster. It's not that that reaction wouldn't happen, it would just take a very, very long time. So enzymes help to speed up chemical reactions by lowering the activation energy. And usually when we talk about an enzyme, remember that it is a protein catalyst, so it is composed of a protein, and usually it ends in A's. So if we were thinking about lactose and being able to break it down, what we can actually use is an enzyme called lactase, and it can break it down into simpler sugars, or even smaller components than that. But we use lactase to break down lactose. You may hear about lipase, which is going to be useful for breaking down fats, or we'll talk about catalase. We'll talk about protease, which helps to break down proteins. Usually the enzymes end in ASE. So if you see ASE at the end, more than likely it's a protein. Now, it's not directly involved in the chemical reaction, so the enzyme can be reused over and over and over again. It just helps to speed up the chemical reaction. That's all it does, is it helps to speed it up. It lowers this activation energy, speeds up the reaction, and it can be used over and over. And enzymes are very tightly regulated as well. because we don't always want enzymes on. So sometimes they have to be tightly regulated because of that. Also, the enzyme is very specific to a particular substrate. So if you look down at this figure below, the enzyme that you see is in purple. So again, that would be your lipase, protease, sucrase, whatever it may be. And what it does is it has to bind to very specific substrate molecules. When it binds those substrate molecules together and they form this complex between them, then what's going to happen is it's going to form a product. So the substrate molecules came together utilizing this enzyme, and then we're able to form a product at the end. But nothing changed to the enzyme. Again, the enzyme is not truly used in the reaction. It just helps to speed it up. And one thing is that, again, the enzyme is very specific to that particular substrate. So it has the ability to recognize the substrate, recognize that shape, form that complex, and therefore make the product that is needed. So definitely make sure you know some characteristics about enzymes. And when we talk about enzymes, we use them quite frequently, but we can have their shape very much regulate their function. So unfortunately, we have to be very careful with an enzyme because there are some factors that can alter the shape of the enzyme, or what we know as the conformation. So usually we use heat to speed up chemical reactions, but if you overheat something and you're using very high temperatures, it can actually do what's called denature the enzyme. And so it basically renders it inactive and doesn't allow it to work. And so it's an irreversible change. It destroys that enzyme and it now no longer holds that shape. If it doesn't hold that shape, it doesn't hold that function. Now, we can also have factors like radiation, electricity, particular chemicals, and even poisons in different pHs also affect that enzyme. And again, what they can do is lead to this denaturation phase, where it's actually inactivating the enzyme, rendering it completely useless. Because again, it changes that shape, therefore it changes the function, and that enzyme does not work correctly. Now, I want to go back to now this idea of what chapter 4 is going to talk about. So we made mention of this idea of actually producing proteins. And so what the DNA is going to do, that deoxyribonucleic acid, is that's going to be holding the genetic material. And we store that genetic material, again, in the nucleus. So when you think about your cell, think about the nucleus and think about that DNA within the nucleus. That DNA stores instructions. It is an instruction manual or a blueprint how to produce certain functions of proteins. So again, how do we make our enzymes? How do we make our blood proteins that are very much needed? How do we produce antibodies and actually make structural parts of our muscle? How do we produce hormones? And how do we make these cellular components? Even when it comes to metabolic capabilities and how tall we are and what enzymes are used to make the pigments of our eye color, all of that information is stored on the DNA. So anything that you physically see about yourself, that phenotype, this physical expression, is because of DNA, because we have this genetic expression. So we're going to talk more about that here in just a moment. Now, to catch you up with some terms, we're first going to think about DNA, and then we're going to start moving into the idea of making proteins. But here are just a few definitions for you. So when we think about genetic information, this is your instruction manual that tells you how to make these proteins. And again, it's stored in a particular DNA sequence, basically a step-by-step direction of how to make that protein. And when we think about DNA, again, we think of this long double helix 2-strand structure. But one gene codes for one protein. And a gene, by definition, is a sequence on the DNA. So you have your DNA, which is sort of larger, and then on the DNA itself, you have particular genes, and they code that information for one protein. So if you needed the protein or the gene to make lipase, you would again look for that one sequence of DNA to make that one protein, that one enzyme. If you needed to construct antibodies, again, you look for that genetic sequence of DNA, that one spot to make that one protein. But when we think about all of the genetic information in a cell, we actually think about the genome. So you may have heard of the Human Genome Project that sequenced all of our genetic material. Because not only do we have DNA found in our nucleus, we also find DNA in our mitochondria as well, and in different areas. So the genome is all of the genetic information that a cell contains. And then like in plant cells, they contain it in their plant cells as well in different areas. Again, not only do they have it in their nuclei, but they also have it within their chloroplast and chlorophyll. So we'll talk more about that probably later on. All right, now within this genome, there are specific proteins that are or specific portions that code for particular proteins. Those are called exomes. I won't be asking you about exomes, so you don't have to worry about that. But the genetic expression that you have, again, controls what proteins are going to be produced. Just because you have all of these genes doesn't mean they're always expressed. And they can be expressed in different amounts or in different circumstances. So that is very tightly regulated. Again, if you don't necessarily you don't have a lot of lactose present in your body, you probably don't have to make a lot of lactase. Versus, something else that we may use quite often, like something that helps us with glucose breakdown, or maybe helping us with breakdown of glycogen. That may be something that we use more frequently. So that might be turned on. So with gene expression, it really is regulating and controlling what proteins are produced, in what volume, and again, under what circumstance. All right, so again, this is just to come back to that idea with DNA. So we're going to go in a little bit further. So now thinking about DNA, we're thinking about what's inside the nucleus of the cell. The DNA is made up of nucleotides. One nucleotide contains a sugar phosphate backbone and this base, which we call a nitrogenous base. For DNA, it's A and T and C and G. Adenine, cytosine, guanine, or thymine. And the DNA is going to have a double strand to it, and it strands and twists into this double helix structure. So want to make sure you feel comfortable with that. And remember that your base pairs, or your base pairs that complement each other and come together to form bonds, are A and T and C and G. So this is what you're going to see here when we think about them coming together and being double stranded. And remember, they're going to be in this double helix structure. All right, now when we start thinking about this, again, the big thing to keep in mind is that when we think about this double strand, those base pairs that you see in between are actually going to have base pairing. We call it complementary. So again, the way I used to remember this is that my name was Alyssa Thomas. So A and T go together and C and G go together. And this just gives you a much better picture of that double helix structure. And the hydrogen bonds are actually what are in between the base pairs that we can see. And when we look at the DNA itself, I love this picture because it's showing you the DNA in sort of this big picture, and then it shows you what happens at the chromosome level. So the DNA actually wraps around these histone proteins in order to form that chromosome, that X that you see at the bottom. Now, just a little brief thing to keep in mind as to why this is so important and why DNA is actually looked at and used in basically forensic science, in criminal investigations. We actually do a lot of DNA and genetic profiling. So the human genome, and again, the genome is all of the genes that you have in your cell, all the genetic information that's in the cell. There is 3.2 billion bits of information. And so when we think about that, we have a particular DNA profile. Each person is going to have their own genetic profile and be a little bit different, especially when you think about their fingerprint. So when we think about DNA profiling, We use it to check for crime scenes. We're able to check for it when there's human remains after a natural disaster. We can check for familial histories as well, especially for paternity tests. Or again, we can establish innocence in crime cases. So for instance, if there's a rape victim, they may be able to take a specimen from that rape victim and be able to find out who the true rapist was using genetic profiling. We use something called gel electrophoresis, which is very commonly done. We won't discuss that in this class, but if you ever take a higher level biology, you'll probably learn a little bit more about it. And as sad as it is, because if you think about, especially before, DNA technology and profiling was a thing, a lot of prisoners were put into, or a lot of innocent people were put into prisons, and they didn't really truly get a fair shot. And it wasn't until, many years later, almost 20 years later, hundreds of prisoners have actually been freed due to genetic profiling, realizing that they actually had the wrong person. And actually, they've actually freed quite a number of people on death row for this manner. So it's definitely a big deal, and it solves these cases. It puts some closure to those families, as well as, again, it actually gets the right person. Now, really quickly, I do want to go into some detail and give you a little bit more of a review. All right, so we talked about the idea of the cell cycle. And in the cell cycle, we talked about the idea of one parent cell breaking into two identical daughter cells. And we had mentioned that the cell cycle was primarily made up of interphase, a little bit of mitosis. Let me fix that. So this little part of mitosis, and then a very small little sliver was that cytokinesis, the actual splitting of the cytoplasm. But primarily the cell spends majority of the time in this interphase. And so in this interphase, we have G1, S, and G2. Hopefully that sounds a little bit familiar. What we're going to start talking about is what happens in this phase here, in the S phase. And what happens in the S phase is we're actually going to duplicate the DNA. So when we talk about DNA replication, think about this every time you make a new epithelial tissue, anytime you make a new skin cell, or anytime you make a new muscle cell. This is what's taking place. You're starting off with one piece of DNA for that one cell, and we have to copy it to make two. So when we talk about this idea of DNA replication, this is going to be happening when the cell divides. So again, you're going to have each daughter cell receive identical pieces of DNA. And so what we do is we produce the exact same copy of DNA. And again, this occurs in the S phase of interphase. So what has to happen, and remember, this is a double helix, double-stranded piece of DNA. Those hydrogen bonds in between our base pairs have to actually break. And then we're going to unwind our DNA. We use a specific enzyme. Again, the enzyme's that nice little biological catalyst, speeds up the reaction. We use something called helicase to unwind the DNA and actually separate it. Then what's going to happen is, now that we have this double-stranded DNA, again, normally in this double helix, we actually unwind it. So now it's two separate copies. What we're going to do is we're going to use it as a template to make new strands. So at the very end of this reaction, now you have two copies of the DNA. One piece you're going to use has one original strand, and then the other strand will be new. And so what we do is we add new nucleotides to that exposed base, and we use an enzyme called DNA polymerase to do this. And now we're going to make our new phosphate sugar backbone connection, form two new pieces of DNA, and therefore we can then continue on with mitosis. So I hope that makes more sense. We're actually doubling up the DNA. Instead of having just our one copy that we had before, now we're going to have two copies. So we have the identical DNA to make two new cells. All right, so I hope this picture gives a little bit of a better idea as to what you are seeing. So remember, this is occurring in the S phase. And what you're going to see again, this is happening in the DNA. We have this DNA replication. So we have this double helix structure, and we're breaking down those hydrogen bonds in between our base pairs. When we break them down, now we have two individual strands, and we use those strands as templates. So that way we can grow, and using that DNA polymerase, add on new base pairs to our new template. So when this is finally completed, instead of having just one copy of the DNA, we have two copies. So this only happens when the cell is going through the cell cycle. So I just wanted to make mention of that. Now, when we think about the DNA itself now we have to think about what happens when it goes to make proteins. So I want you to think about it, that DNA replication doesn't mean that it's going to go for protein synthesis. This is just to copy that DNA. But when we think about now making our amino acids and then having our proteins actually be built, we need to have a specific genetic code. And the genetic code is going to be a sequence of 3 nucleotides that we call the triplet code. And that 3 nucleotides, whether it be ACG or TGA or AUG, whatever it may be, that represents an amino acid. And it's going to signal either the beginning or the end of the protein. So that triplet code, based upon the sequence, dictates what amino acid is going to be used. And that amino acid, again, is going to create long strings. And so you'll have string of amino acids that will form a particular structure for a protein. That structure greatly relates to its function. And so the DNA itself actually stores this master copy of the genetic code, and it remains in the nucleus. So your DNA is always going to be found within the nucleus. But the protein synthesis that occurs actually occurs in the cytoplasm, and it occurs on the ribosomes in the cytoplasm. If you remember that we talked about ribosomes helping us to make proteins, this is where that information becomes so important. So RNA is also going to be used to make proteins. It copies that genetic information that we got, and it transfers that information from DNA in the nucleus to the actual cytoplasm. So it conveys a message that's usable in order to send it to the ribosomes. So therefore we can actually start to develop and make these proteins. All right, so when we think about RNA, it's a little bit different from DNA. So as I said before, double-stranded is going to be DNA, single-stranded is going to be RNA. And each nucleotide for RNA is going to be very similar than it was to DNA. We have adenine, guanine, and cytosine. But instead of using thymine, we use uracil instead. So when we think about complementary base pairing, now instead of it being A and T, C and G, now we use A and U. And because this RNA is only going to be a specific segment of the DNA to make that one protein, it's much shorter than the DNA. And so what allows us to go from DNA to RNA, to actually transcribe the RNA and allow us to convey this message in a way that makes sense, to actually make our amino acids, we go through something called transcription, this actual copying the DNA into an RNA sequence. And in more detail, we actually use messenger RNA to make that happen. It carries the genetic code from the DNA to the actual ribosome. And what allows us to actually form this RNA in the proper strand is using this enzyme called RNA polymerase. So again, if you see the ASE at the end, it's an enzyme, biological catalyst, speeds up those chemical reactions. All right, so when we think about transcription, we're using that RNA polymerase to actually recognize that strand of DNA. Because remember, we're looking for one gene to make this one protein. So we don't need to copy the whole piece of DNA. We just have to copy this one little spot. That's the one spot we're transcribing. And so it's a section of DNA that's going to unwind and expose that template that we need for the coding. Then what's going to happen is those nucleotides, the complementary nucleotides, actually pair with the DNA bases. So instead of having thymine like we did for DNA, we actually use uracil. So really quickly, let me show you kind of an idea behind this. All right, so really quickly, let's do a little recap on replication, and then we'll talk about transcription. So that way we don't get these confused. Remember, DNA replication happens during the cell cycle, and we're copying the whole genetic material. So when we think about our DNA template, we're going from one strand of DNA and making another strand. So if this was our piece that we were copying, now our message is going to be based upon that complementary base pair. So here we would have A, C, because we'd make the complement to this. Remember, A and T go together, and C and G go together. So then here we'd have G, G, T, A, G, C. That's for DNA replication. So you're just copying the DNA. Now in transcription, what we're trying to do is we're trying to go from DNA to RNA. And when we do this, we're actually going to messenger RNA. So now we have A and U, C and G. So now our DNA molecule, let's just say hypothetically, again, we had this little piece here. We had T, T, G, C, C, A, T, C, G. That's our DNA molecule. And now we're going to make and transcribe our messenger RNA. And we're going to form those base pairs. So now what we have is A, C, G, G, and here we normally have T, but because instead of having thymine, we use uracil instead. And then we'll have A, G, C. So this is now going to be our new piece of RNA. And remember, we show it in threes because those threes are our codon. And that codon is going to give you the representation for one amino acid. So I hope that makes a little bit more sense when we start talking about these differences. All right, so when we're thinking about this protein synthesis, again, we start off with our DNA, and we're going to transcribe it into RNA. And again, in more detail, we transcribe it to messenger RNA. And then finally, what's going to happen is imagine, if you will, that the DNA is your genetic code, your genetic blueprint, but maybe it's in a different language that doesn't necessarily make sense. So what we do is we transcribe it into RNA in order to have it make sense and convey this message. And now once it's in a readable term, now we can truly understand it and then go on to truly make our protein. And so going from DNA to RNA, we call transcription. Going from RNA to our protein, we call translation. So be careful with these two, transcription versus translation. Transcription occurs in the nucleus. Translation occurs in the cytoplasm, and more importantly, on a ribosome. So I hope that makes a little bit more sense when you're trying to keep all these concepts together of what in the heck is going on. I know it can be a little bit confusing. So this is to help us make protein. Versus when we copy the DNA, again, that's to make a new cell. So just be careful with those two concepts being sort of different. And even though we talk about them in the same chapter, kind of compartmentalize them into being two separate pieces. And that will sort of help you out to know these differences. All right, so talking more about those transcription pieces, again, we use this enzyme called RNA polymerase. And it's recognizing the specific strand of DNA because remember, we're looking for one gene. And that one gene codes for one protein. So we don't need to copy the whole, we don't need to transcribe the whole piece of DNA. We're just transcribing this one gene to help us make this one protein. So that certain section unwinds, exposes that genetic code. And then again, those complementary base pairs can actually go in and start forming this new messenger RNA piece. And it will continue until it finally hits a signal. It's called the termination signal, and that's at the end of the gene. Once you've hit that, now that mRNA strand is released, and remember, this is happening in the nucleus. So the DNA rewinds and goes back to the double helix structure. And now your mRNA strand is going to leave the nucleus to attach to a ribosome within our cytoplasm. So hopefully that makes a little bit more sense as to why we had to learn about ribosomes and cytoplasms and rough ER. They all kind of now are coming back to be very useful and help us understand these concepts. All right, so again, remember with DNA that it's double-stranded. Remember, it's deoxyribose, and you have the base pairs A and T, C and G. With RNA, again, this is going to be single-stranded. It's the ribose sugar, and we have A and U, C and G. DNA is going to be our genetic blueprint. RNA is this readable conveyed message to allow us to actually go make that protein. And so when we start talking about this idea of our amino acid sequence, remember that each amino acid is specified by a particular sequence of 3 bases. So if you had TTA, this is called a codon. And this one codon represents one particular amino acid. And if you had AAG or GCA, this is a codon, this is a codon. They all specify a particular amino acid. And when we string them together, this is what allows us to actually start producing our protein. And so when we think about now the translation that takes place, now we have this messenger RNA that we got from the nucleus, and it's going to attach to a ribosome. So the mRNA leaves the nucleus, binds to a ribosome, and acts as this template to actually start making the protein. And at the ribosome, this genetic code that's carried by this messenger RNA is actually used to start manufacturing or synthesizing this protein. So in translation now, we're converting the genetic code into these amino acid sequences. And again, if you think about a protein, we know that that is the primary sequence. And then we can go on to do the secondary, tertiary, and then finally we'll get this quaternary structure. So this is sort of how it all unfolds together. So here's a nice overview of sort of what is going on. So it's showing you this idea of your DNA, then how it goes to actually producing that messenger RNA, the messenger RNA leaving the nucleus and actually attaching to the ribosome, and how it's truly making this strand of amino acids come together. And eventually, again, when we get this string of amino acids, we call it a polypeptide chain. Once this translation is done, we'll have this very long polypeptide chain. And then what will actually be done is then it will go to the rough endoplasmic reticulum to actually convey the true shape. Again, then it will start going into that, that secondary structure using hydrogen bonding and twisting and pleating. It'll hit that tertiary structure and start actually forming like a 3D piece. And then finally, we'll get that quaternary structure that truly forms the shape of our protein. All right, so thinking about protein synthesis, it requires that your amino acids are in a proper sequence. And when they're attached together and they grow this polypeptide chain, they have to have a proper sequence in order to make this happen. So transfer RNA, or what we know as tRNA, aligns the amino acids during this protein synthesis. With the tRNA, it binds onto the amino acid. And when it does this, it actually is going to bind onto the mRNA according to the sequence. And when it does this, it adds on an amino acid to the growing polypeptide chain. So when you go to see it here, this is actually what we're looking at. This is the transfer RNA. And as you can see, it's coming into contact with our messenger RNA down below. When it comes on, it allows to have this amino acid come and actually attach, and they grow and grow and grow and grow. And that's kind of what we can see going on down here. So that's why it's sort of referred to as tRNA, or this transfer RNA. And then again, as the ribosome starts to move down the mRNA, the tRNA brings another amino acid and another and another and another and another, and it grows this polypeptide chain into our protein. So when we talk about the amino acids, though, there are 20 potential types of amino acids you can see, and there are 64 possible codons that can take place. So depending on what codon you use, it all depends on the sequence as to what the structure will look like, and therefore, again, what the actual function will look like. But when we talk about truly allowing for this to take place, this translation and protein synthesis, it has to start at what's called the initiation codon. So this AUG actually starts the signaling to start making this protein. And it continues growing these amino acids in these polypeptide chains until it finally hits a stop codon. And once it hits the stop codon, that is the end of this protein. This is the end of that amino acid sequence. And they don't have any corresponding tRNAs to go along with it. So for each mRNA codon, there is a transponding, or a corresponding, transfer RNA, and we call it the anticodon. So when you see them come together, there's the codon from the RNA, and then there's the anticodon from the tRNA, and they come together to correspond to allow for this linking up and actually allowing the amino acids to grow. So this is just a picture of what you can see with those different types of amino acids. You will not be asked to memorize them in any way, shape, or form. Just want to show you what happens when you have these codons and how they code for these particular amino acids. And again, there are 20 potential amino acids you can come up with, and 64 different outcomes of how they can be organized. Now, what you're seeing here is actually showing you that mRNA molecule on the ribosome. The brown portion is the ribosome, and the transfer RNA is actually bringing the amino acid to the ribosome and allowing for this attachment to take place. And so again, for each codon in your mRNA sequence, you have a corresponding anticodon for that transfer RNA. And so when we think about protein synthesis, it actually occurs on the ribosomes in the nucleus. The mRNA is the template for protein synthesis, and that tRNA brings the amino acid to the ribosome, binds it to the mRNA, and allows this amino acid chain to continue to grow. Now, when we think about protein synthesis, as I said before, we think about ribosomes. They are the ones that help us to actually compose the protein or make the protein. And there's different parts of it that are made up in these protein molecules. They're called rRNA, or ribosomal RNA. And it's basically just two subunits that come together. But when we have this association with the ribosome, that's when you have the binding of tRNA and mRNA. And again, the tRNA brings the amino acid to the ribosome. The mRNA serves as the template. And so the more tRNAs you have come to the area, when they come together, it's going to grow this polypeptide chain. And the amino acids are, again, joined by that peptide bond. As I showed you guys in the review, when we seen that carboxyl group with that amine group came together, it had that bond in between. That's how these are joining, and they're joining and joining and joining and joining. And then finally, once the ribosome hits that stop codon, the protein is released, and we are good to go to actually move on with this protein to get the proper folding, proper shape, 3D structure, send it to the rough-ER again to make sure it's properly formed, and then it can go out to the Golgi. All right, so just kind of as a breakdown, this is the idea of what happens in transcription and what is going to occur in translation. Now, when we think about this, again, all of us have our genetic information. And for the most part, 99.9% of our genome sequence is the same. So we are very, very similar. But that 0.1% of our genome that varies is going to make us have these unique characteristics. So this could be what gives us different appearances. Why I have blonde hair and hazel eyes, why my sister has more of a lighter brown hair and has green eyes, why my mother has super blonde hair and super green eyes, or why my dad had dark hair and blue eyes, what made my dad so tall versus me being only 5'7" and my sister being 5'5". Or what gives the medical effects to us that affects our health, like having genetic probabilities for Parkinson's or ALS, or the genetic sequence to have the BRCA1 or BRCA2 gene. That 0.1% of the genome that varies really creates these unique properties. So again, we can see things that affect our appearance. We can see things that affect our health. And we also have some variations that we actually don't see observable effects to, meaning that there is a change, but we just don't physically see that appearance change. So this is what brings us into the idea of mutations. So a mutation is a change to that DNA sequence. So remember that from a DNA sequence, we think about those base pairs, AATT, CC, GACA, whatever it may be. If you change that DNA sequence, it's called a mutation. And so sometimes this occurs when you add out a base pair, you delete a base pair, or maybe you even swap a base pair for another one. And this can be spontaneous, where it just happens and there's really no control from it. It just inserts a base pair, or it can be induced. And this is what we think about really now in a more of a problematic situation, because this is due to exposures from mutagens. So our chemicals that we think about are exposure to smoke and inhalation and smog and harmful radiation. And again, certain additional growth hormones that are added and these chemicals that can actually induce a mutation. So they are called mutagens, and it's an induced mutation that does it. And down here on this side, again, we can just see how this is a change. So the reason we like to show this one is this is actually what happens with sickle cell patients. It's a change of one base pair that completely destroys the shape of the red blood cell and leaves the red blood cell a sickled manner, which is how it gets its name, sickle cell anemia. So that change from T to A, which normally that T coded for glutamic acid, now actually codes for valine. And again, because now we made a change to our DNA sequence, when we went to go make our amino acids, that changed. So it changed the structure, therefore it changed the function. And again, it left a mutation that is actually quite detrimental. Now, when we start actually talking about mutations, not all mutations are harmful. Actually, there are some that are quite beneficial. I shouldn't say some, and it really happens, but there are a few mutations that actually do have a very positive effect on health. So for instance, there's a mutation that some people have against the HIV virus, where the virus actually can't bind onto the receptor properly, and therefore they are not afflicted with HIV or then being able to be afflicted with AIDS. But for the most part, when we think about mutations and their effect on health, usually it's a non-functional or a missing protein. So as I said before, sickle cell anemia is a great example. So is Duchenne's muscular dystrophy. And when I teach patho classes, I love to talk about Duchenne's muscular dystrophy. We actually had a good friend that passed away from this at 20. Usually it's more affected in boys, and they don't tend to see 21. But Duchenne's muscular dystrophy, it's a mutation in the gene that codes for something called dystrophin. And because of this, it actually causes muscle cells to start to not function correctly. And so they start to break down, they start to have a lot of wear, and it results in very severe muscle weakness to the point where again, you may have a normal functioning toddler, but by age 3, they start having issues with extreme muscle weakness, fatigue. They may actually be in a wheelchair, and eventually it starts to cause problems with their respiratory system. And usually they pass from respiratory distress. So just something to keep in mind, big picture as to why we talk about this. Now, there are some ways that we can protect ourselves from these mutations. So our DNA itself actually has a repair enzyme that corrects to see if we have any of these mismatched nucleotides. And so it can try to fix it. If it's possible, it's going to try to fix it. Now, when we think about the nature of the genetic code, usually there's 2 to 4 codons that actually are going to produce the same amino acid. So if you had a simple mutation where it was one of the 2, it doesn't affect the protein. So that's actually okay, and that's not necessarily a problem. And also what we can do is have 2 copies of that chromosome as well. So if one copy is mutated, the other copy may actually be enough normal function that we maintain health and we don't actually see these big picture issues at the end and cause, you know, sickness or infection to occur. All right, that's all I have for you for this class. I hope you enjoyed this lecture, and I will see you guys very soon. Bye.